Good morning for everyone. Today I'm gonna present to you the PhD research that is named Antibody Immobilization in Thin Thin Film Transistor for Low-Cost Biosensor Applications. I am Rafael Salinas and I work at the Applied Biotechnology Research Center from the National Polytechnic Institute, Mexico. The abstract of this work is the following. As an introduction, I will talk to you about the food safety and the great importance that this has acquired along the years for different countries and all over the world. This issue, joined to the excessive population growth, has a big impact on storage, processing, and transport of foods. They in some cases ends up in a foodborne disease. The World Health Organization are very clear. 600 million of people get sick every year, and from them are reported 420,000 deaths associated mainly to 31 agents, such as a virus, toxins, parasites, chemical products, and bacteria. The bacteria commonly related to this disease are Salmonella, Campylobacter, E. coli, Listeria, and Vibrio cholerae, found then spread all over the world. Here I present a schematic representation about the geographic distribution of disease. Mexico and USA are found in the American bloc A and B. The statistics of worldwide burden of foodborne disease assigned uh, the pathogens assigned to our country are mainly associated to enteropathogenic E. coli, enterotoxigenic E. coli, norovirus, Campylobacter, and Salmonella. The principal group of risk are older adults, immunocompromised people, and toddlers. In the last 20 years, pathogen detection has been focused on the food industry, the clinical sector, water and environment, among others, being Salmonella and E. coli the species better investigated. Immunoassays, biochemical and molecular techniques such as polymerase chain reaction are the most used for pathogenic detection, given high detection levels. However, and the need to develop technologies that can help these methods in order to reduce reagents, cost, and processing times for vulnerable sectors is of high importance. Since the last years, a new field study has been developed consistent on biosensor devices. A biosensor is an integrated receptor transducer device that uses a biological recognition element to provide selective quantitative and semi-quantitative information. In this schematic view of biosensor, I present the biological recognition element that can be an enzyme, an antibody, DNA, or protein, whose can be attached to diverse supports and their biochemical change can be transduced through different systems for signal processing. The proposed strategy in this work is the use of ZnO-Thin film transistor, whose active layer acts as support for the biological recognition element. Here present a schematic view of the general components of a biosensor. For the results and discussion, there are different challenges in the development of biosensors. However, the biosensor design with eco-friendly perspective will have a great projection in the near future. Devices on recyclable plastic substrates compatible with flexible electronics could be the next step for the development of low-cost and easy-handled clinical biosensors. In this work, we use the inverted coplanar structure or bottom gate because it's the one allows to work with the biological recognition element over the active layer. The observed components of these transistors are polyethylene terephthalate or PET, indium thin oxide, silicon oxide, the contacts drain and source, and the active layer of zinc oxide. Since the transistor fabrication was developed at low temperature, 200 Celsius degrees, on a recyclable material, this turned them in an eco-friendly devices that conserve their electronic properties. And the biological recognition element used of this work are polyclonal antibodies against enteropathogenic E. coli. The biofunctionalization strategy consists of the hydroxyl groups generation, the functionalization, and finally the antibody immobilization. The use of silane at the functionalization stage provides an available amine group to react with carboxyl groups of the antibody's heavy chain to form an amide bond. In previous works have proved the antibody mobilization in zinc-oxide thin films, and this method was adapted to be compatible with thin film transistor to assemble biosensors on plastic substrates as PET. In order to observe the electrical behavior of these biosensors and the detection signals measurements of the transfer characteristic curves were performed. But the needed to a validation method is present in this work, and the Bradford method used is based on the specific bound between the glucose and amino acid from proteins. Using this quality, it is possible to quick and efficiently observe a color change in the thin film transistors that have the antibody immobilization in the zinc oxide thin films transistor, and this is that you can see in this figure. The electrical characterization of transistor without biological treatment is shown as the negative control. It is observed that when the antibodies are immobilized, the transference curve has a displacement that is related to the contribution of antibodies at the conduction channel of the device. Then, when the bacterial detection takes place, it is observed a new contribution that modify against the conduction channel, and it is suggested that the membrane lipopolysaccharides and phospholipids from attached bacteria have a resistive effect that causes a high voltage requirement to reach the threshold voltage. These quantified signals are quite important because uh, for the development of biosensor with an electrical transduction response, the observed displacement isn't in function of the analyte concentration, and that is in concordance with other authors. The results obtained demonstrate the functionality of low-cost materials for the development of biosensors. The use of plastic substrates and their processing at low temperature offers an advantage for their fabrication in diverse applications, and that is advantage. As a conclusion, we have the methodological process for the immobilization of antibodies has been found to be reproducible. The process of the chemical modification of the transistor active layer does not affect the electrical behavior and or any of the components, which allows a transfer curve to be obtained and their process conditions are reproducible. It is worth mentioning that the use of plastic substrates and the processing at low temperature enables the development of immunosensor devices with an important projection for the low cost and sustainable technologies. This is our acknowledgement and thank you very much for your attention.